Thank you very much for the introduction and also for the opportunity to present and to discuss some of the work, recent and ongoing work we have been doing on this topic that has been funded mainly by USDA through collaborative agreements, also by the Center for Outcomes Research and Epidemiology at Kansas State University, and our current projects are being funded by the Swine Health Information Center. So as demonstrated by the introduction of the West Nile virus, the Zika virus, and the chikungunya virus in the American continent, the North American continent specifically, United States is a region that is suitable for the introduction of other vector-borne diseases. So what is it about Japanese encephalitis virus, or JEV for short, that is specifically of concern to us in United States? So one of the first concerns is how JEV has and is continually expanding to other areas of the world, as demonstrated by the a local or autochthonous human case that was reported in Angola in 2016. Also, the identification of genetic material in birds and in mosquitoes in northern Italy, although there are no human cases to date. And most recently, the expansion to areas southern of the Torres Strait Islands in Australia, specifically to the eastern and southeastern states in Australia, as we have learned from our Australian colleagues. So in addition to the continuous expansion of JEV globally, we have competent hosts and vectors in the United States. While considering what we define as the wild or the sylvatic transmission cycle, which is characterized primarily by birds as the primary host and mosquitoes, there are over 90 species of birds that have been identified as competent for JEV, so they can become infected and transmit JEV, with water birds being one of the most susceptible. Of importance is the fact that responses and susceptibility to JEV infection, the genotypes 1 and 3, were determined in several North American bird species, and it was deemed to be highly variable after experimental inoculation in studies that were conducted by Nemeth and Bowen, doctors Nemeth and Bowen in Colorado State University in 2012. Within what we consider the domestic transmission cycle, pigs act as amplifying hosts. And in fact, not only domestic pig, but also feral pigs are considered important JEV reservoirs, basically or primarily given the high viremia titers, the fact that Culex mosquitoes, which are the primary vectors, they feed on pigs in nature, and also because of the high turnover rate in the pig industry, which allows the constant or the maintenance of a high number of susceptible pigs. To complicate things even further, as we have described, as we have discussed in the previous presentations, feral pigs are also important JEV maintenance hosts. And they have been reported in at least 35 states in US, and they are rapidly expanding given their high adaptability to a variety of climates and conditions, and also the fact that they do not have natural predators, among other factors. What is also of concern, despite the evidence of vector-free transmission, vector-mediated transmission is still the most efficient. There have been several species of Culex, mosquitoes of the Culex genus, as being identified as the most competent vectors for JEV. And of those, Culex tritaniorhynchus is the most important competent vector in Southeast Asia, followed by, as we have learned, Culex annulorostris and Culex gelidus in Australia. There have been more than 10 species of Culex mosquitoes that have been deemed competent for JEV. And of those, it's important to know that we have found Culex tarsalis, Culic and Culic as also competent for JEV, and those are present in United States. Of importance is Culic kingifaciatus. Researchers at the Biosecurity Research Institute at Kansas State University demonstrated the competence of Culic kingifaciatus mosquitoes after oral exposure with genotype 3 strains. And that work was done here in United States. Among other factors that are of concern is specific to US is globalization and the increase in travel to and from Asia, as well as the increase in trade. Also, we have climatic and environmental conditions that are very similar to the ones that exist in epidemic countries. There is cross-reactivity, as we have learned, with other flaviviruses, which makes diagnostic very challenging. Also, currently, we do not have an active surveillance program for JEV in United States, and our preparedness and response plan is a little bit outdated, so it needs some update in order to, again, mainly updated with the new or most recent information that has been produced, at least in the past 5 years or so. And lastly, the inability of the public health agencies to prevent those outbreaks, and also the further establishment of diseases such as West Nile after its introduction in 1999, make US a region that is suitable for the introduction, for the potential introduction of the Japanese encephalitis virus. So what we did is, a few years ago, actually in 2013, we were approached by the United States Department of Agriculture, ARS. It used to be at the time the arthropod-borne animal disease research unit. Now it's the foreign arthropod-borne disease unit. So they, part of their CREASE project, they were looking at investigating more about Japanese encephalitis and Rift Valley fever, among other emerging diseases. And so they approached us to conduct an epidemiological evaluation of looking at the risk of introduction of Japanese encephalitis in the US. So with that task at hand, we put together a team. Our original team consisted of, was a multidisciplinary, multi-institution group of researchers. And currently our group has expanded and includes collaborators from what is called now the Foreign Arthropod-Borne Animal Disease Research Unit, also from USDA APHIS, the Wildlife Services, and also include colleagues, faculty, graduate and undergraduate students from the College of Veterinary Medicine at Kansas State University. Our objective was to quantify the risk of introduction of Japanese encephalitis virus in the United States. To quantify the risk, we decided to conduct or to use a risk assessment approach that what it does is determines what can go wrong. So what is the hazard we are interested in? So how likely is that hazard to occur? What is the probability? How can it happen? So what are the main drivers, as well as what are the main consequences or impact of that hazard? And we put all that information, and we considered all that information over time and under specific circumstances. So there are different types of risk assessment approaches. Some are considered qualitative, quantitative, and there are semi-quantitative. And that depends, or the choice of those depends on the type of source materials as well as the data available. So in order to answer this particular question, we use qualitative and quantitative approaches. So in a brief summary, so qualitative approaches are basically consist of non-numeric, or so there are risk categories that are not necessarily numerical, they're categorical. And so to depict risk levels. And then we also have quantitative approaches, by which we use mathematical models in order to obtain a numerical expression of risk. So we started by compiling evidence from the literature. So we compiled evidence on competent hosts and vectors present mainly in United States. as well as potential pathways of introduction of the virus in the United States, and other environmental factors that are relevant to introduction and transmission of Japanese encephalitis virus. And we integrated all that information into a qualitative, we initially conducted a qualitative risk assessment approach. So for this particular approach, we use an already validated framework that was specifically designed to assess the risk of vector-borne livestock diseases, and it consists of a structured questionnaire, where the answers to those questions can be done in the form of risk categories, it can be descriptive statements, or yes or no scenarios. And then we also proceeded to use a quantitative risk assessment that I'm going to describe after. So for the qualitative risk assessment, so it consists of two main stages. One is hazard identification, and second is the risk assessment. For hazard identification, first we start by describing the pathogen. So what are the main characteristics of the Japanese encephalitis virus? What are competent vectors, host, their distribution, abundance, and the risk areas? Specifically for our model, we looked at, we defined the area at risk to be continental US. Therefore, we excluded Hawaii and other US islands and territories. And we evaluated, we assessed risk over time and space. And the risk specifically pertain or refer to both the present and the future risk. When looking at the risk assessment part is subdivided into 6 different subsections that it consists of the probability, describing the probability of entry, of transmission, probability of establishment, extent of spread, likelihood of persistent, and the impact of disease to then obtain a risk estimate. In order to characterize the probability of entry or of introduction of JEV in the US, we assess 7 main pathways of introduction, which consisted of, first, the introduction via infected vectors, either via aircraft, cargo ships, tires, or wind, and those can be in the form of eggs, larvae, or adults. Also, we looked at the entry via imported live animals, entry of migratory birds, imported biological materials, imported animal products, important contaminated biological materials, specifically vaccines, as well as the entry of infected humans. Based on the available evidence of all the pathways of introduction that we considered, only the 2 were deemed non-negligible, and those were via the introduction of infected vectors, specifically adult mosquitoes, via aircraft and cargo ships. And the introduction via aircraft was deemed to be very high, which is the highest risk category that we could consider. Then that was followed by the introduction of also adult mosquitoes via cargo ships. And for that particular pathway, the risk was deemed from low to moderate. In our risk assessment, the importation via live animals was considered negligible. And we understand that that may be considered an important pathway for other countries. And so in this case, specifically, it was considered negligible because there is no legal importation of live pigs from JEV affected countries to United States. And also there was some anecdotal evidence on illegal importation of live pigs. And there was no information about movement of live animals, especially zoo, show animals, or other animals of the Suidae family. And those were all considered.
Apologize. Can you minimize the image there on the right, please? Thank you. I'm sorry to interrupt.
No, that's fine. Is that better? Yep. Okay, thank you. All right. Then also the other pathways of introduction through migratory birds, biological materials, animal products, vaccines, and humans were all deemed negligible based on the information we had available at the time. And there were three main factors for that. One is either because some of the aspects were not relevant, that were they were trying to, some of the aspects that were explaining some of these pathways were not relevant to JEV introduction and then how it transmits. Also because some of them were very complex to be plausible, or because the mechanisms of occurrence were not known. In terms of the probability, should I? Yeah, is that better? Okay. I learned that in a class. A student told me that, and I was like, I had that annoying man there forever. Anyways, now I know. So now we're looking at the probability of transmission. So in summary, probability of introduction was considered very high based on the introduction via adult mosquitoes via aircraft, and then low to moderate via the introduction again of adult mosquitoes via cargo ships. Now we're moving into the probability of transmission. So the probability of transmission, which refers to the probability that the virus is able to spread to susceptible hosts, was considered from low to high risk, so highly variable, depending on the vector distribution and the host density across the country, and depended on the season and the US state. And it was specifically considered or deemed low, of low risk, around airports and seaports which were deemed as the most important ports of entry of JEV in the United States. So after considering all the factors related to the probability of transmission and deeming it to be low to high, then we proceeded to evaluate the probability of establishment. The probability of establishment referred to the probability that the virus can spread from vector to host and then vice versa. Okay, so we deem the probability of establishment to be negligible. And that was based on the fact that there was low availability of amplifying hosts, mainly around airports and seaports, again, the main ports of entry, based on our previous determination of probability of introduction. Also, given the very short duration of viremia and amplifying hosts, which is up to last up to 3 to 4 days. Also, given the low contact rates among competent hosts and vectors, potential cross-protection with other flaviviruses, especially of West Nile virus and St. Louis encephalitis that are present in the United States, and also because of the high environmental liability of this virus. This virus is liable to UV light, gamma radiation, disinfectants, among others. So the probability of establishment was deemed negligible. So we stopped our qualitative risk assessment at that particular stage, considering the JEV establishment in the US is compromised. So it was deemed again negligible. Because we found that the probability of introduction of Japanese encephalitis in the continental US was low to moderate in cargo ship and very high in aircraft, we decided to then quantify the risk using now a quantitative risk assessment approach. So what you see here is our conceptual model. And basically what we were interested in quantifying was the probability that at least one infected adult mosquito will enter the United States either via aircraft or cargo ship from the different JEV affected countries into US in a certain at-risk period, which was deemed 9 months from March to October for the aircraft model, and all year round for the cargo ship. So we built 2 models, one for aircraft and one for cargo ship. So as you can see, some of the variables that we use to parameterize this model, which is a stochastic model, some of the parameters were considered were the sensitivity of the detection method to capture mosquitoes, the number of mosquitoes that were found or not found, either again in aircrafts or cargo ship. We look at also the number of aircrafts that were put at risk period or cargo ship coming from the JEV affected areas to United States, as well as mosquito infection rates. The data that we considered was mainly data that we obtained from 2010 to 2016. We considered only port-to-port vessels. We assume in our models that mosquitoes can survive the Trans-Pacific ship crossing. And also we assume that the number of mosquitoes that we can find in cargo flights are the same as the ones that have been identified in passenger flights. Considering the JEV affected countries, we divide them in 9 different regions based on latitude, whether they were epidemic or endemic, as well as were based on the mosquito infection rates. So they were divided in 9 different regions. So what we found is that we have a very high probability of introduction of at least one infected adult mosquito per year. And so the median probability of introduction of at least one infected mosquito is 95%. And again, the median number of infected mosquitoes that enter via aircraft every year to the US is 3. Out of those regions that were considered endemic or epidemic for JEV, Eastern China was the region that had the highest probability of introduction with a median probability of 0.7. And on the median number of mosquitoes that enter from Eastern China every year was calculated to be 209. And of those, a median number of 1 with a range of 0 to 4 are actually infected, that JEV infected. So that is in terms of where are these mosquitoes, infected mosquitoes are coming from. Now, where do they get? So in the United States, so where are they mainly or most likely introduced? So we found that is the Mediterranean, California. So what we did, we divided US, our continental US, based on the ecosystems, the ecological regions of North America. So it's basically this division takes into consideration the different ecosystems, air, land, water, and other biotic factors. And it was considered that the area at the highest risk of introduction, again, of one infected mosquito via aircraft was the Mediterranean, California, which is this area that is depicted here in light green. Followed by the Eastern temperate forest, which is pretty much represent all the East Coast. When considering now our second model, the cargo ship model, we consider that the risk of introduction of at least one infected mosquito from JEV affected regions into US via cargo ship was negligible. Some of the factors that we did not consider at the time included, for instance, recent evidence on vector-free transmission, which we learned from the reclaim studies, where there was some inoculation, some IBE inoculation of pigs, and they show evidence of pig-to-pig transmission. So we did not consider vector-free transmission in our models. There were some other data that were not available, where it was very limited. For instance, the distribution of feral pigs and also their potential impact in the introduction and transmission. And there were other factors that we considered they had to be reassessed. So what we are doing currently is reassessing some of those pathways, pathways of introduction, and then also we are putting a lot of effort into trying to determine the role of pigs, not only domestic, but feral pigs in the transmission of JEV in US and what kind of impacts that's going to have. One of the areas we're currently working is on redefining the regions of introduction, the potential areas of distribution in the United States. We wanted to add a little bit of more granularity. So rather than saying they're going to be potentially the risk of introduction is higher in a particular ecosystem, we also are incorporating not only information on the ecosystem, so environmental and other important biotic factors into these regions, but also we want to incorporate, or we are incorporating, the distribution and density of pig production and also of feral pigs. So what it means is some of the regions that we are not going to be considering, then what we're going to do next is we are going to be looking at evaluating the risk of introduction and also transmission in each of those regions, because we wanted to incorporate some of those nuisances that may be present at the regional level, in which we may have dismissed when looking at US as a whole, right? In terms of looking at continental US as a whole. So we are going to consider specifically the risk of introduction by each of those regions, as again, considering distribution of host, domestic, and feral pigs. We're going to be now considering also flights from these states in Australia where they are presently having outbreaks. We are also going to be looking at incorporating recent evidence that we have been compiling from 2016 and on. There have been, fortunately, lots of new papers and new literature on the subject, especially modeling papers looking also at transmission, and specifically how the vector-free transmission may impact the other routes of transmissions that are known. In terms of transmission, we are incorporating information on host density, vector abundance, climatic and regional factors, and specifically we are looking into the role of pigs and domestic and feral swine, again, in transmission in general. And we are using that, we're collecting that from peer review literature, from data from collaborators, and we have formed an advisory group that we're going to use to elicit expert opinion. So based on what we found, our models, based on the literature and the data that was available at the time, we consider that JEV can enter every year via adult mosquitoes, via aircraft, and less likely via cargo ship, but mainly via aircrafts. And then there is potentially transmission, but there is no establishment. The conditions are not suitable for establishment of Japanese encephalitis in US, Japanese encephalitis virus in US. However, as we have learned, as conditions change from population growth or movement, increased animal movement as well, especially of other species or other amplifying or reservoir hosts such as feral pigs, migratory birds, and specifically the effect of climate events, we need to understand that we have to, and that's what we are doing right now, revisit some of the pathways, because we may have to change some of our model assumptions, and that may result in some changes in the probability estimation. Several of the what's next, there are several knowledge gaps that are still remain that we need to elucidate. So I'm very looking forward to the rest of our discussions on this topic. Some of the areas that need more or further exploration. So I'm going to leave it as is right now. And with that, I would like to thank again the funding and all our collaborators and my colleagues at Kansas State for all the work, and obviously all of you for your attention. Applause. Thank you. Megan.
Natalia, thank you for the presentation and great job. So the pig-to-pig transmission is somewhat poorly understood, I suppose, a good way to put it. I've seen it in the literature where it says that component of transmission is considered non-negligible, but we heard from our Australian colleagues that their perception of pig-to-pig transmission was non-existent in their experience. Can you tell us a little bit more about what's the recent research shown for pig-to-pig transmission, and your take on, is there a difference with genotype 4 versus other genotypes?
So so your first question regarding evidence and whether the role of direct or vector-free transmission, how important it is just in general in transmission of JEV. So there hasn't been a lot of other additional studies that have explored the vector-free transmission, except for what we have seen with the Ricklin paper in 2016. There is some work also done at the Biosecurity Research Institute at Kansas State University, where they also inoculated pigs. They did an intravascular, so an IV inoculation. And so except for that, there is not much out there. And I'm not sure whether that is publication bias, where there may have been other studies, and then because they didn't obtain a certain type of results that were not published, or there may be that they're going to be published in recent years. I know that some of our colleagues at FABADRU, they are actually they're starting pig studies this year. In fact, maybe they're starting in the next couple of weeks, and they were planning to start 2 years ago looking at pig studies, transmission studies. However, because of COVID, that had to be delayed. So we have these colleagues, but obviously they're in very, very early stages. But other than that, there is no much there. So that is in terms of the actual experimental studies looking at vector-free transmission, not much there. There are some transmission models that have been done recently. There is one, I was talking to Dr. Hills earlier, there is one study from Diallo, I think it's there from Cirado of the Institute Pasteur, and they look specifically at, they compile or they consider serological data, I believe was from Cambodia, and they look at how much vector-free transmission impacted transmission in general. So they compared, they had transmission compartmental mathematical models or differential equation models where they look at vector-free transmission and vector-free and vector-mediated transmission. And what they saw is that there is a more than a 30% impact of the effect of vector-free transmission on the basic reproduction ratio. So it seems on mathematical models, and that was consistent with the serological data from pigs that they collected. So based on that, it seems that there is a potential impact, but they are not seeing, as we have learned from our colleagues, that effect in the field. And it may be because, and that's one of the questions I had, did they collect samples in all animals, or they just look at animals with clinical signs? And so I think we still don't know enough. There is not enough evidence, but it's, when you compare, I think something that we have been doing a lot is comparing what the little we know about Japanese encephalitis with what we have learned about West Nile virus, which is fairly similar. It's a flavivirus of the Japanese encephalitis group. So there are lots of parallelisms, and in West Nile virus, as well as in other flaviviruses, you see that there is host-to-host transmission. It's still not the most efficient. Vector-borne is the most efficient for all flavivirus, and that's why we consider them as vector-borne diseases in general. Does that make sense?
Yeah, no, that helps. Thank you.
No, thank you.
We have a question from a virtual attendee, Dana Cole asks, I'm sorry I didn't follow why migratory birds were eliminated as a pathway. Was the prevalence of the virus in endemic countries deemed too low, the relevant species non-migratory to North America, or infection too transitory in birds?
That's a good question. Okay, I'll try to. Thank you for the question. That's very good. I had it because I think it's a very important pathway of introduction, especially after what we learned that probably the entry via migratory birds may be one of the most important pathways of introduction for the recent outbreaks in Australia, in Eastern and Southeastern Australia. So the reason why we deem the entry via migratory birds as negligible was because, and again, in summary, all the events that could lead to an introduction via migratory birds was too complex to be plausible. What that means is, so we don't have a direct flyway, migratory flyway from Asia to United States. And these are the main two flyways that are of relevance to us. the West Pacific flyway, and the East Asian Australasian flyway. So they all, the migratory flyways go from south to north, right? And then so the main ones that the migratory birds flying from Asia mainly come from these two flyways. So where we could see potential transmission and introduction was potentially in Alaska, where there will be a connection, the intercontinent connection of flyways, where you have these two flyways coming from Asia, and then we have these two main flyways that go south, and those are the Pacific Americas and the Mississippi Americas flyway. So for us to see introduction, we will have to have a bird, migratory bird coming from Asia in either of those two flyways, meet a competent vector, a mosquito vector, and then potentially infect another migratory bird going from Australia and taking again the Pacific Americas on the Mississippi Americas and going south, okay? So there are a few things here that play against all those mechanisms. Here, we are looking at a fairly long distance. It is considered that migratory birds take between 3 and 5 days to fly from the Korean Peninsula to potentially Alaska. And that we are right in the actual period in which we could see viremia. So it has to, that migratory bird will have to find a competent mosquito in Alaska. What happened in Alaska, we only have 7 weeks where mosquitoes are active, right, due to the cold temperatures and the latitude. And there are very few species of competent vectors in Alaska. Only one is mainly considered competent, and that is the Aedes vexans, which is not a Culis, is not deemed as competent as the other species that we talked about. And then we will have to have that the next migratory birds that was infected by the competent vector in Australia to go south, and likely will be introducing again on the West Coast. And so those are some of the factors that make it very, very hard for this process to happen. The other potential wave, for instance, will be through Hawaii. However, when the flyways, when migratory birds go through Hawaii, they're already going south to then move again north and go back to Asia. So they will be leaving United States by the time they go through Hawaii. Yes.
So another question from a virtual attendee, a good follow-up question to what you've just answered. This comes from Daniel Peach of UGA in the Department of Infectious Diseases. He says, does the spread of JEV vector mosquitoes northward and the disproportionate warming of northern areas change your risk assessment of introduction via migratory birds? For example, from infected East Asian birds getting lost and ending up in Alaska, Yukon, or British Columbia. He recently finished a postdoc in British Columbia, he says, and we were finding JEV vector species like Culex pipiens spreading into northern British Columbia and Culex tarsalis in Yukon territory, although it may have been present all along and undetected. Should this be a concern for JEV introduction?
One of the things that I mentioned is that, again, when you assess these pathways, it's based on the available or the limited data that you have. And so at the time, there was no really available data from Canada or Mexico as it pertains to JEV. So that is one. So that is a potential risk, specifically from contiguous countries. The climatic change that we didn't consider, and we recognize that that's a limitation, and that's what our Australian colleagues are seeing. So absolutely, as the climate changes, as there is change in the temperatures, and there is an expansion of mosquitoes, we may identify other potential vectors that are competent for JEV. Absolutely, those are important pathways to take into consideration. Yes.
Just a little more on the birds, because you mentioned Hawaii. So given there is that movement into Hawaii, and there's Quinquina fasciatus in Hawaii. I'm not sure how many pigs there are in Hawaii. Given, excuse me, Hawaii is part of the US, is there a risk for importation and transmission in Hawaii that's greater than the continental US?
So we did not take into consideration Hawaii. So we only consider continental US. And so I cannot speak about that because we actually did not evaluate. What I can tell you is there is not a lot of information in terms of potential JEV surveillance in Hawaii in general, because there is no surveillance in the continental US. There is no such surveillance. I'm not aware of surveillance for other RV viruses in Hawaii that we can use to understand the situation there. So that will be very interested. But in terms of we did look in terms of the migratory pathways, the potential impact of Hawaii into continental US. But then again, we deemed it not relevant because of what I mentioned before. But I'm not aware of efforts that have been done in Hawaii. I know that there is zero. I need to clarify now that I remember. So Dr. Vienna Brown with Wildlife Services, they had been collecting feral pig sera in the last, so there is surveillance that has been done. They have some data, seroprevalence for Japanese encephalitis in feral pigs from Guam, and they have data that they collected. They have samples from 2021 and 2022 from different states in US, as well as from Hawaii and Puerto Rico, and they are in the process of testing those samples for Japanese encephalitis. So I think we're going to learn about that, hopefully in recent months. I believe she may be presented in October. I'm not sure if that's the Australian conference that they mentioned on wild birds. She may be presented. She was talking to Dr. Bowen at Colorado State University to test a subset of samples. And they were using the plague reduction neutralization test to evaluate. I got a thumbs up, so that's the test to test those samples for presence of JEV. So I think that will be a very, very important information for us. Some of the other states, Dr. Michael, maybe you know, but I believe they're looking at serum samples from also Texas, California, and Florida, I believe, in addition to Puerto Rico and Hawaii. I don't know if you recall.
No, as far as wildlife services activities outside of Guam, I can speak to some of their feral swine surveillance, but as far as specific to JEV, I don't know that I can comment on that. But if you don't mind, I can share some numbers for Guam to kind of extrapolate on what you were saying. So the data that I have is from FY14 to FY18, and there were 600 samples collected. Of those, it looks like there were 5 positives and 4 suspects, both found in FY17, and they represented about 2.7% of the samples collected. Ignore that last part. I apologize. So 5 and 4, so 5 positives, 4 suspects out of the 600 samples collected in the 5-year period of time.
Yes, thank you.